Okay, so welcome to this next video in which we are discussing the function of synaptotagmin. Okay, so far what we've done is we've discussed the formation of the core snare complexes, the trans-core snare complexes strictly, and how that leads to the docking of synaptic vesicles at the presynaptic membrane. We now want to look at the function of synaptotagmin. We want to look at what causes these vesicles to fuse with the plasma membrane when an action potential arrives. Okay, right. So, when an action potential propagates along this membrane, it causes a depolarization of that membrane. Okay, so the electrical potential difference across the membrane is going to depolarize. There are channels in the membrane of this axon terminal which can detect that change in electrical potential difference and will open in response to it. And the ones which are important to us, which I'll label here, are voltage-gated calcium channels. Voltage-gated calcium channels. Now, voltage-gated calcium channels, there are many different types of voltage-gated calcium channel. And the sort of voltage-gated calcium channels that are in the membrane of the axon terminal like this, of neurons, are what are known as N-type and PQ-type voltage-gated calcium channels. So I want to try and explain to you, what does N-type and PQ-type actually mean? Well, voltage-gated calcium channels are made up of multiple subunits. The main and most important subunit is a subunit known as the alpha-1 subunit, which is the one I've drawn here. It's the one that actually has the pore which allows the calcium to move through. So it's the one which actually is a calcium channel. The others all just stick on the side of it and modulate its function. Okay, now, there are 10 genes in the human genome which code for alpha-1 subunits, and they all are slightly different. They all have slightly different organic-based sequences. They lead to slightly different amino acid sequences when you actually translate them into protein, but they do the same thing almost. To say that the voltage-gated calcium channels in axon terminals are N or PQ type means that you're picking specific genes to make these alpha-1 subunits. So if you are an N-type voltage-gated calcium channel, it means that you have used the CAV2.2 gene in order to make the alpha-1 subunit of your voltage-gated calcium channel. If you are PQ type, it means that you have used the CAV2.1 gene in order to make the alpha-1 subunit of your voltage-gated calcium channel. Okay, now, you bind onto this alpha-1 subunit, as I alluded to earlier, many different auxiliary subunits, which I'll draw here. So these are the accessory or auxiliary subunits. So I'll colour them in as well. So in purple here, sitting alongside the alpha-1 subunit in the plasma membrane, is what's known as the gamma subunit. This subunit down here in the cytoplasm is the beta subunit, which we'll colour in orange. Okay, this subunit over here, which I'll colour in turquoise, is the alpha-2 delta subunit. So this is alpha-2 delta here. Okay, and the reason it's called alpha-2 delta is it consists of two subunits, in fact. The alpha-2 is the box at the top, and the delta is the stick, which basically is suspended in the membrane there. Okay, right, so that's the overall structure of a voltage-gated calcium channel. When the membrane electrical potential difference is depolarized by an action potential arriving in the axon terminal, this voltage-gated calcium channel will open, and it will allow calcium to move in. The calcium gradient across the membrane is absolutely enormous. So calcium concentration in the extracellular space is about 1.5 millimolar. And calcium concentration in the cytoplasm is 100 nanomolar. So it's around 15,000 times greater outside. So there is absolutely no question of what direction calcium is going to move in. It's going to move into the cell when you open this channel. Okay, now, the alpha-2 delta subunit It's responsible for positioning these voltage-gated calcium channels so that they are nearby the synaptic vesicles, which are docked to the membrane, basically. So that when you open the voltage-gated calcium channel and allow calcium to enter the cytoplasm, you effectively spray the machinery on this docked synaptic vesicle with calcium. Okay, so calcium is going to go up in the vicinity of these docked vesicles. Now, there is a protein in the membrane of these docked vesicles, which I'm going to draw here, okay, awkwardly. Right, so how am I going to do this? It's got a membrane-spanning alpha helix, which is implanted into the membrane of the synaptic vesicle, and then it's got these two special domains in the cytoplasm of the cell. Okay, and it's the star of the show. It's what this video is about. This is synaptotagmin here. So let me colour this in. So this protein in pink here, this is synaptotagmin. So let me just label this up. So this is synaptotagmin. Right, okay, so it has these two domains. The one that's closer to the membrane-spanning alpha helix is the C2A domain. So let me draw it again down here. Okay, so here's our synaptotagmin again in the membrane here. So this is the C2A domain, and this is the C2B domain. And both these domains bind calcium, basically. So the C2A domain binds free calcium ions. So free calcium ions are going to bind to the C2A domain. And the C2B domain binds two calcium ions. Okay? And when these calcium ions bind to the C2A and C2B domains, it does not cause a change in conformation in the C2A or C2B domains. Instead, what it causes is a change in the electrical potential of those C2A and C2B domains. So the electrical potential of the C2A and the C2B domains goes up in response to adding these divalent cations onto it. Now, what's going to happen is that the synaptotagmin is now going to interact with many things in this plasma membrane, okay? One of the things is that it binds to syntaxin 1, okay? Another thing is that this C2A domain is going to bind to a component of the phospholipid bilayer known as phosphatidylserine, okay? And finally, the other thing is that the C2B domain is going to bind to a component of the phospholipid bilayer known as phosphatidylinositol 4,5-bisphosphate. So let's go over the way and discuss this in a bit more detail. Okay, right. So, if this is our synaptic vesicle here, then we've got our synaptotagmin here now, and I'll draw these two domains like so. Okay, I've drawn them bent over so that I can draw them now both interacting with this membrane. Okay, now in the phospholipid bilayer, there are a lot of phospholipids, but there are also modified phospholipids. So let me discuss the modifications that you can make to phospholipids. So, if we draw the phospholipid bilayer horizontally here, then the usual constituents of the phospholipid bilayer are phospholipids. So let me draw the structure of a phospholipid in cartoon form anyway. Okay, so this is the cartoon for a phospholipid here. Okay, and these sort of tails here, which I'll draw in orange, those are the long-chain carboxylic acid tails, or the hydrophobic tails. So basically, in a phospholipid, you have a glycerol backbone, and then you have a sterified two long-chain carboxylic acids, or fatty acids as they can also be called, long-chain carboxylic acids. You've esterified these long-chain carboxylic acids to the first and the second hydroxyl groups of the glycerol molecule, which is this horizontal molecule here in turquoise. Okay, so this is glycerol here, horizontally. So you esterify two long-chain carboxylic acids to the first and the second hydroxyl groups of the glycerol molecule, and then to the third of hydroxyl group of the glycerol molecule, you bind a phosphate group through a phosphoester bond. So this is a phosphate group. Now, this is the structure of a normal plain old phospholipid. So this is a phospholipid, okay? Or the old name for a phospholipid is to call it a phosphatidate molecule. And this is going to help us, phosphatidate molecule. This is going to help us understand what phosphatidylserine and phosphatidylinositol 4,5-bisphosphate are. So, we'll begin with phosphatidylserine then. So basically, let me write its name out in full. So phosphatidylserine, basically what you take is you take a phosphatidate molecule and you bind it to a serine molecule. So you just stick this phosphatidyl group onto a serine molecule, or if you want to do it the other way around, you stick a serine molecule onto a phosphatidate molecule. So let me draw this here. So, if this is our phosphatidate molecule again, what you're going to do is I'll draw the serine as a triangle here, like so. So this blue triangle, as I'm going to colour it in, so this is the blue triangle here, this represents a serine molecule bound through its hydroxyl group to the free hydroxyl group of this phosphate group via a phosphoester bond. Okay, so you bind the R group of the serine amino acid to the phosphate group of this phosphatidate molecule. Okay, and I realise that I'm colouring these in the wrong colours. So I've coloured that in green, but it should have been in turquoise. So here's turquoise. Okay, and then finally orange for these long-chain carboxylic acids. So basically, phosphatidylserine is just a modified phospholipid. It's just a phospholipid with an extra bit stuck on its head. Similarly, PIP2, okay, as it's all usually denoted, which in full stands for phosphatidylinositol. Phosphatidylinositol 4,5-bisphosphate. 4,5-bisphosphate. This is just a modified phospholipid. It's just a phospholipid with an extra bit stuck on its head, basically. So let me show you this. So again, we'll draw out the structure of our normal phospholipids. So here are the long-chain carboxylic acids, the glycerol and the phosphate group. So let me colour those in. So glycerol was in turquoise here. Okay. The phosphate group was in red here, and the long-chain carboxylic acids were in orange here. Okay, now, if we want to make phosphatidylinositol, then we need to link this to inositol this time. Now, inositol is a 6-carbon ring where all of the bonds between the carbons in the ring are single bonds, okay? And every single one of the carbons in this ring has a hydroxyl group coming off it, and also a hydrogen coming off it. So it's an incredibly rotationally symmetric molecule. Now, what I've made so far, that is phosphatidylinositol. I've linked the phosphatidate group onto an inositol molecule. To convert this into phosphatidylinositol 4,5-bisphosphate, I have to add 2 phosphate groups onto the 4th and the 5th carbon of this inositol ring. So this is the 1st carbon, this is the 2nd, 3rd, 4th, so I add a phosphate group on there, 5th, so I add a phosphate group on there. And I bind these to the hydroxyl groups that come off all the carbons of inositol through phosphate ester bonds or phosphoester bonds. So this now is the structure of phosphatidylinositol 4,5-bisphosphate, and I hope you can again see that it's just a modified phospholipid. And we'll continue this discussion in the next video.